Hi everyone, my name is Vienna Protomastro. Today's date is May 11th, 2024, and today I will be doing a presentation on the regulation of sperm maturation in the germinal epithelium. Alright, so first off, let's review some of the general structure of the male reproductive system. The male reproductive system consists of the testes, the internal genitalia, which is the accessory glands and the ducts, and the external genitalia. The external genitalia consists of the penis and the scrotum. The scrotum is the external sac into which the testes descend so that they can stay cooler than body temperature. The urethra, located right here in this diagram, serves as a passageway for sperm and urine and runs through the ventral aspect of the shaft of the penis. The urethra is surrounded by the corpus spongiosum, which is a spongy column of tissue. The corpus spongiosum and two columns of tissue called the corpora cavernosa make up the erectile tissue of the penis. And the tip of the penis is enlarged into a region called the glans, which at birth is covered by a layer of skin called the foreskin or the prepuce. And the male accessory glands, the internal genitalia that we were talking about before, include the prostate gland, the seminal vesicles, and the bulbourethral or Cooper's glands. The bulbourethral glands and seminal vesicles empty their secretions into the urethra through ducts. The individual glands of the prostate open directly into the urethral lumen. Okay, so the human testes are paired ovoid structures that have a tough outer fibrous capsule that encloses masses of seminiferous tubules, which is the region of the testes where sperm and hormones are produced. The seminiferous tubules leave the testes and join the epididymis, which is where sperm will complete their maturation. The epididymis becomes the vas deferens, which is a tube that carries sperm from the epididymis to the urethra, where it will exit into the external environment. Okay, and so the seminiferous tubules, again, are the site of sperm production and contain two very important types of cells, the Sertoli cells and the interstitial or Leydig cells. The role of the Sertoli cells is to regulate sperm development by providing nourishment for developing spermatogonia. They manufacture and secrete proteins such as inhibin, activin, growth factors, enzymes, and androgen binding protein to support sperm production. Androgen binding protein is secreted into the seminiferous lumen where it will bind to testosterone. And then the interstitial or Leydig cells secrete testosterone and first become active in the fetus when testosterone is needed for development of male characteristics. And the seminiferous tubules also have three main compartments, the lumen, the basal compartment, and the interstitial fluid. And we can see pretty much all of these components labeled really nicely here in this diagram from our Silverthorne textbook. Okay, and so spermatogenesis can be defined as the process in the testes where spermatogonia divide and differentiate through mitosis and meiosis to produce mature sperm cells, or more commonly known as spermatozoa. In the spermatogonial phase, spermatogonia, which are diploid germ cells, undergo mitotic division to produce primary spermatocytes. In meiosis 1, primary spermatocytes undergo the first mitotic division, resulting in the formation of secondary spermatocytes. Secondary spermatocytes undergo the second meiotic division to produce haploid spermatids. In spermiogenesis, the spermatids undergo a series of structural and morphological changes to become mature spermatozoa. And then the mature spermatozoa will then detach from Sertoli cells and are released into the lumen of the seminiferous tubules. These spermatozoa will then move towards the epididymis for further maturation and storage before ejaculation into the external environment. Alright, and so there are quite a few hormones involved in regulating spermatogenesis. FSH, or follicle-stimulating hormone, stimulates Sertoli cells in the testes to support spermatogenesis by promoting germ cell proliferation and differentiation. LH, or luteinizing hormone, stimulates Leydig cells in the testes to produce testosterone, which is essential for the maturation and function of germ cells during spermatogenesis. Testosterone, produced by Leydig cells, acts on Sertoli cells and germ cells to produce spermatogenesis and maintain reproductive function. GnRH, or gonadotropin-releasing hormone, is made in the hypothalamus and regulates the synthesis and secretion of FSH and LH from the anterior pituitary gland. And then lastly, inhibin, produced by Sertoli cells, serves as a negative feedback regulator of FSH secretion, helping to maintain optimal levels of FSH for spermatogenesis. Alright, so here is a quick study aid that I made that might be useful for our upcoming final exam when reviewing the male reproductive physiology section. If you'd like to pause the video to try and write the letter of the correct match next to each problem, you can feel free to do so. Alright, and so here are the answers to the study aid. The seminiferous tubules, again, are where sperm is produced. The vas deferens is what carries sperm from the epididymis to the urethra. The epididymis is where sperm completes its maturation. The interstitial cells secrete testosterone. The Sertoli cells provide nourishment for developing spermatogonia. And lastly, inhibin serves as a negative feedback regulator of FSH, as we talked about in one of the previous slides. Alright, and that is the end of my presentation. Thank you so much for watching, and good luck on the final exam. Have a great rest of your night.